Most of it really is fine. It's
Okay. Hello, everybody. Happy week, last week of classes. Aren't you glad?
yeah.
Yeah. Okay, so immunoassays. Today we're going to talk a little bit about immunoassays, give examples of them and applications. So when you hear immunoassays, what does it mean to you? Can you give me anything that you might know about immunoassays? Go for it.
Are they for checking for allergens?
Yes, they can be used to check allergens. All right, great. What is the basis of these assays? What do we use in these assays?
Electricity?
We use electricity? In what sense, Isaac? I am
Well,
not
I see
sure.
that thing up there. It looks like it has.
Oh, use voltage.
Yeah.
And we definitely need electricity to hook it in. I like that creative answer. Okay, but what else do we know about amino acids? When you, what is immuno? What does it remind you of? Yeah, what's in the immune system? What works? Antibodies, antigen and antibodies. So basically the allergy that we talked about is that when we are exposed to an antigen and our body doesn't like it and recognize it as foreign matter, there are B cells in our body that will produce antibodies toward this antigen. The next exposure to this allergen, we're going to have an allergic reaction. Okay. So how we utilize this outside of the body? Outside of the body in analysis, we utilize this by having antigens and antibodies in one assay and detect the presence of antigen or quantitate the antigens in the assay. Antigens can be proteins that are allergenic. Antigens can be proteins that are GMO, if we want to detect presence of genetically modified organisms. Antigens can be toxins. Antigens can be anything, basically. Mostly proteins, but they can be non-proteins. They can be mycotoxin, for example. They can be pesticide. They can be anything. And we utilize the antibodies to detect the presence of these antigens. So this is kind of a brief overview of what immunoassays are, utilizing antibodies and antigens for detection. So, what are antibodies? Antibodies are actually proteins themselves. So here is an antibody, immunoglobulin. Immunoglobulin is made up of 4 polypeptides or 4 proteins, heavy chain, 2 heavy chain, those are the bigger proteins. That's why they're called heavy chain, and they're identical. And then light chain, they're also identical. And then you have this Y shape. And here is a region to bind, so binding site. So every antibody has 2 binding sites. And this is the antigen. The region on the antigen that binds to the binding site, what's it called? Does anybody know? I don't have it here, maybe on the next slide. But that region that binds to the antibody is known as epitope. So the region of the antigen that binds to the antibody is called epitope. So it's good to know that term, epitope. So their proteins, they're produced by B cells in response to an antigen. And they have a very strong interaction with the antigen. So antibody-antigen interaction is among the strongest interaction that we can have that are non-covalent. So non-covalent interaction can be ionic, hydrogen bonding, hydrophobic, van der Waals, no covalent reaction, no disulfide linkages, nothing covalent. So non-covalent interaction, but the strongest interaction there are. They have very high affinity. The antibodies have very high affinity to the antigen with a very high affinity constant. So this is kind of a real kind of structure of an immunoglobulin. So you see that yellow here is one peptide, the light blue is another one, the dark blue and the green are those light chain peptides. So the epitopes is the region where it binds to the antibody. So two things you want to remember: linear epitope, conformational epitope. This is very specific to a protein antigen. Okay, this is specific to a protein antigen. So proteins are made up of what? What are the building blocks of proteins? Amino acids. So the epitope can be linear, where you have here 1, 2, 3, 4, 5. These are linear amino acids. This can be an epitope that binds here. So this is the red is a sequence of amino acids that bind to an antigen, and where they bind is the epitope, linear epitope. It can be conformational. You know, the protein bends and folds. So the way they fold, there might be 3 amino acids here and 2 amino acids there. When they are in close proximity to each other, they form an epitope that an antigen can recognize. So these are nonlinear conformational epitopes. So as the protein folds. So I want you to remember linear conformational epitope. Another thing on this slide is I want you to remember that there are antibodies that are polyclonal and antibodies that are monoclonal. Now you're going to say, why do I care? You care because in standard immunoassays that you purchase, like ELISA, immunosorbent enzyme linked. Now I'm losing my words, but anyway, ELISA, I will show it in the next slide. In monoclonal is very important. It's as standard reagents. I want to show you what I mean by monoclonal and polyclonal. Illustrated. All right, so let's say this is a protein. Okay, you have an epitope here. So this is going to bind to the region on the antibody. Okay, so monoclonal, that means all of the antibodies that you have in your reagent are going to bind to this region, all of them. So they are the same antibodies. They bind the same epitope. Polyclonal is what we actually have in our body. So if we're allergic to an antigen, a protein, our bodies, we have multiple B cells. We have many B cells. Every B cell is going to generate a different antibody. So I'm going to have potentially an epitope here, an antibody for this region, an antibody for this region, an antibody for this region, for example. So these are polyclonal. Same antigen, but they bind at multiple epitopes. So polyclonal, we get that from our serum, from a human serum or animal serum that has allergy, for example, to an antigen. They would have multiple B cells. Different types of B cells would generate different antibodies to different region on that protein. And every person is different and every animal is different. You have a variety of immunoreactivity, we call it. So we have a variety of antibodies. Monoclonal, what they did, scientists figured out that if they get one type, let's say X B cell, if they get one type of B cell, they can culture it if they link it to a cancer cell that can proliferate fast. So what they did is they linked a B cell, one kind of B cell, to a cancerous cell, and then they can culture B cells, same kind of B cells, then they can get a monoclonal antibody. Again, why is this important? It's very important to buy monoclonal antibodies for an assay. Standard. It's a standard antibody that you can buy for your assays. Makes assays easier to work with. Okay. So, characteristics of immunoassays. So the characteristics of immunoassays, they're very specific. So very specific to the type of antigen we want to test. So that's great. They have high sensitivity. That means we can detect differences at very small concentrations. And then very simple. There are standards. You can buy the agents for immunoassays, and it's very simple to do. So, but the immunoassay requires that we have the correct antibody for the antigen of interest. That's a requirement. Another requirement, we need to be able to differentiate a bound antigen, that means an antigen that is bound, versus a free antigen in the system. It's very important, free, bound. And I'll let you know later on why this is important, because it's important for detection purposes. So we need to differentiate between bound and non-bound antigen. And there's another feature for proteins. Antibodies are proteins. We rely on their hydrophobic surfaces to bind them to our plates where we do the analysis. We do them in microplates. I don't know if you've ever seen a microplate, but I'll show you a picture of it. It is a plastic mostly. It has hydrophobic surface, so the protein can bind to the surface of the plate via hydrophobic interaction, and that's very important for these types of assays. How do we produce antibodies specific for measuring the targeted antigen? From a biological standpoint, you have an antigen and you want to get antibodies. How do we get antibodies? From an animal. We don't always have serum from human to do this type of work. Sometimes we do, sometimes we don't. So if we want to go say, I have a rabbit, what do I do to this poor rabbit or poor goat? Not that I like it, but that's what they do. So,
Just a guess, but you have to expose it to the antigen. So
that is a great guess, a correct guess. You just inject the poor animal with the antigen directly to the blood, and then their B cells are going to generate antibodies. Then we take the blood and we isolate the antibody. We are very cruel humans, but this is how we learn, and this is how we become better in medical fields. Okay. We talked about some applications. We talked about allergens. We talked about GMO as an example, detecting presence of genetically modified organisms or species. Another one is common in meat species authentication. Did you hear about a few years ago there was horse meat in the burger? Yeah. That's how you can find if you have horse meat or something that is not supposed to be in your burger. Supposed to be beef, and then you found horse meat or you found pork meat in your beef. So you would use antibodies specific to a protein that is only found in horse, that you can detect whether that meat has different type of species than what you thought it is. Toxins, we're going to give an example about aflatoxin, identification of bacteria and viruses is another application. So we're going to learn about, okay, that's where my words, I lost them. Enzyme linked immunosorbent assay, ELISA. Enzyme linked, so we use an enzyme, we link it to either an antigen or to an antibody, and we utilize that in an assay to detect the presence of the antigen. So we're going to learn today about sandwich ELISA. Have you heard about ELISA? In which class? Biology, microbiology. Okay, great. So we have different types of ELISAs. We have the sandwich ELISA, most common to use. We have the indirect ELISA, which is a sandwich ELISA, but I'll tell you why it's indirect. So there is a direct sandwich ELISA and there's an indirect sandwich ELISA, and I will explain the difference. And then we have competitive ELISA, which is very specific to small antigens, non-protein, small antigens, like mycotoxin, for example. So small particle, small antigen, small molecular weight. So in an exam, the final, you're probably going to get a question on immunoassay. If I tell you, give me an example for competitive ELISA, if you say allergen, you're going to lose 5 points. You don't want to do that. So you want to remember the first two are mostly for large antigens, proteins, and the competitive is for small molecules like toxins. Okay. And we're going to talk about other types of immuno-based assay. We're going to talk about, well, we did talk about affinity chromatography. We're going to remind you about that. It's mostly used for purification, either purification of an antibody or a purification of a very specific protein. Lateral flow strip, they are very common. Some of you might or might not yet be familiar, but the pregnancy test is based on lateral flow immunoassay. So I will explain that later. Western blot I have here, where we use electricity, I think.
Our rapid COVID is the same as lateral flow too.
Oh, yes, it's exactly, yes, absolutely. Very good example. All right. So the enzyme-linked immunoassays, we use enzymes for detection. They're easy to work with, they're fast, they act very quickly. We don't have a lot of time when we're doing assays in the lab. We need really some fast methods of analysis. So enzymes work really fast in a short period of time we can get results. So the most common enzymes used in this assay, you don't have to remember them, just there FYI. Horseradish peroxidase and alkaline phosphate. You don't have to remember, but you want to remember that you use enzymes. Why? They are stable. They are proteins, so they can easily be linked to your antibodies or your antigen. They are fast. They catalyze quickly a change. So you can see by spectroscopy, you can get a color and you can detect that color by measuring absorption. So we use a specific substrate that generate the color. Now, plate reader is a very important analytical tool in these assays. Yes, Ash.
What do you mean by easily linked to?
Easily linked because you want to link the enzyme because it's enzyme linked. So you want to covalently link that enzyme to your antibody or antigen. And then, because then you don't want them to be lost during the assay. So that's what it meant, enzyme link, that they're bound, basically, to your antigen or your antibody. So this is a plate, I should have gotten a plate, it's just a small plate, and then you have 96 wells, where you put your reagents in, and I'll show you how we put the reagents and the order of the reagents. And then you allow the reaction to occur, then you would put the whole entire plate in the spectrophotometer and you get simultaneous reading in every well. So you measure the absorption in every well simultaneously. So you can have your standards, you can have your sample, you can have your replication, all in one well instead of putting one tube at a time. So that's very important tool in immunoassays, especially ELISA. So here is a illustration, the drawing of direct detection versus indirect. In this case, the direct detection is your immuno or the antibody that is specific to your antigen is directly labeled with an enzyme. So you label the antibody that is specific to the antigen. The indirect detection, and then you're going to ask them, why do we do that? I'll tell you. But here is where it gets a little bit confusing. You don't have that drawing in your slide? You do? Yeah. So this is an antibody to the antibody. You're going to say, why do I have an antibody to the antibody? I'll tell you what. This is the monoclonal one. This one, you can buy a lot of it. You can get it pre-labeled. You don't have to do the labeling or the linking in the lab. You can buy antibodies that are already linked to the enzyme. And this antibody, the antigen to it is the antibody that detects the antigen. Okay, confusing? You have antigen, you have antibody. In this case, the blue is the antigen to the purple. They're proteins all active. Okay. So why we do that? How do we do that? Is we can get antibodies to a human antibody. For example, if I'm detecting, if I'm running an ELISA for allergenicity to peanut or to soy protein, I have the soy protein or the peanut protein. I have antibodies polyclonal from a human that donated their serum. And then instead of me having an extra analytical step in the lab to label this polyclonal antibody, I can buy a monoclonal antibody already labeled that is an antibody to human antibodies. So the way we do that is we can inject an animal with a human antibody, and then the animal will generate antibodies to human antibodies. And then we can get them purified, and then we can label them, and they become as reagents that you buy. So therefore, it makes the assay in the lab easier. And that's why we have direct and indirect assays. Okay. Is that good? Antibody to the antibody. That's why it gets confusing. Remember, this is protein, so it can be an antigen inside. All right. Okay, so how do we do the assay? So I'm going to draw all these steps visually so that you can see them. Because we don't have a lab on this, but it's nice to visualize. So you have your microplate, you have wells. So I'm just going to say this is a very enlarged base of your well. This is plastic or hydrophobic surface. So this is the base, the bottom of your well in a microplate. So then let's say I'm doing sandwich ELISA. Then I would have my antibodies first. I would put in my antibody and I allow time for the antibodies to bind. They don't always bind nicely like that. Some of them bind uselessly like that, because the protein can bind anyway once. So you don't care about those. You care about here you have the sites where they bind. Okay, so I have my first reagent is the one that contains my first antibody. This we call capture antibody. It will be written on one of your other slides. Capture antibody. Okay. Then what I do is I wash. Every time I put the reagent, I have a wash step. So that's what it says, intermediate washing steps. I wash. So anything not bound will be washed off. If anything, any antibodies that didn't bind, you wash it off. And then you have a lot of open surface. You have open surface. If I put my antigen next, what's going to happen? My antigen, let's say it's a protein. So it's going to bind here, bind here. This we call, if the protein binds here, that means non-specific binding. Do we want that? We don't want that. So I add a block. After I put my antibodies and allow them to interact, I add a block. The block usually is a protein that we don't care for. So bovine serum albumin or gelatin, very common proteins used. So they block any open surface. So they don't bind to your antibodies because they're not specific. They're not the antigen we're looking for. After that, I have a wash again. To wash off any protein that didn't bind. So every time you have intermediate wash step. Then what do I put next? Just antigen, right? Then I put my sample. I put my sample that may or may not contain my antigen. So I'm trying to detect, does my sample have the antigen or it doesn't have? And if it does have it, how much of it is there? So I now put my sample. So on the top here, I'm going to move this way, sample. So then I have my antigen that will potentially bind. Okay. And different shapes for different components. So very specific to my protein of interest or my antigen of interest. All right, and then I wash. To wash off anything that didn't bind. Now what? Let's say if it's sandwich ELISA, I put antibodies again. Sorry, not lambda. I put more antibodies. So now I have antibodies sandwiching etc. So now either I label this with the enzyme or I have another antibody here that is labeled with the enzyme. That's indirect, that's direct. And every time I wash, then I put the substrate, wash, allow to react, then I will have colored product and I will measure the absorption. Okay, so what you want to remember is what we call this, your hydrophobic surface, captured antibody. This is your detection antibody. And then enzyme. Then you add your substrate. And then you will have a product to measure. Okay, so you want to know the different components in the assay. The hydrophobic surface, the capture antibody, your antigen. Don't forget your antigen. Your detection antibody that is labeled with the enzyme, your substrates, your product. Any questions? Okay. Here it is. So here it's showing you that the light blue is your capture antibody. Then this one is your enzyme linked or enzyme conjugated detection antibody. This is the end. There are assays you might come across. We call them butylated. Why? Because you have the biotin. This is a biotin. And then we have streptavidin that binds strongly to biotin. And we label that streptavidin by enzymes. If we do this, we amplify the sensitivity. So instead of having one enzyme linked, we can use this assay where you have biotin and you have the streptavidin that binds to biotin that is labeled with multiple enzymes. You have multiple enzymes on one antibody now. You multiply the sensitivity of your assay and enhance. So sometimes your allergen is present at really very low concentration and you want to detect that, especially if you are looking for potential allergens for labeling. So you really want to intensify the sensitivity of the assay. So this is a very common technique to intensify or enhance the sensitivity of the assay. This is a drawing of the sandwich ELISA. Again, you have your hydrophobic surface. You have your capture antibody. You have the sample that contains many things, potentially including your antigen. And then you have your labeled antibody that is the detection antibody, the enzyme. Then you put the substrate, then you get the product, you measure the absorption. Just basically showing you how we label. So you want to remember the labeling of the different components. This is showing you the indirect ELISA. So again, same principle, you have your antibodies, you have the antigen. You can, let's say it is soy protein in human. We did this research before. We had human sera. We put the human sera of the antibody, you get the antigen. We have another layer of human sera with antibody specific to the antigen. You see, they bind at different epitopes. They're polyclonal. So you have multiple epitopes where two different antibodies can bind. Because you have a big protein molecule, you have multiple epitopes. And then you have the antibody to the antibody. And this is the one that is labeled, and that's the one that you can buy. So you can eliminate the step of doing the binding yourself in the lab and just buy an antibody that is already labeled, and it's an antibody to human antibody in the same time. Okay. So recap for you to think about when you study. The comparative ELISA. So in comparative ELISA, the molecule, the antigen is very, very small. Because it's very small, less than 5 kb or less than 5,000 grams per minute, it's a very small molecule. When you inject it in an animal, it's too small for the B cells to recognize to produce antibodies. But you want to detect this toxin or this molecule. You want to detect that. So what you do, you link this antigen to a protein, bigger molecule. So you link it, and then you inject it in the animal to give you antibodies specific to that antigen. So when you link your molecule of interest to a protein, this is called hapten. So basically you have your protein, and this is your molecule of interest. Together, this is called hapten. This is what you inject into the animal, and in the sera, then you will have antibodies that are specific to this foreign body, as well as antibodies that you don't care for, to the hapten, to the protein. But you really care for this one that binds to your molecule. The whole molecule is the epitope. Because you cannot sandwich that anymore, because you only have one epitope, so you cannot do a sandwich ELISA. Because your epitope is too small. So it has one epitope, the whole molecule binds to your antibody. So that's what it is here is showing. It's called competitive because what you do is you have your molecule of interest, and then you have the molecule that is linked to an enzyme. So this molecule, same molecule as yours, but it's linked to an enzyme. They are competing to binding to the antibody. So that's why it's called competitive. Now, in this case, when you have higher absorption, that means you have low concentration of the antibody. So opposite. Why? Because if you have low concentration of the antigen, more labeled molecules are going to bind to your antibody. So with more labeled molecules, you'll get higher absorption, more color generated, higher absorption. If you have in your sample a lot of your antigen, then less labeled ones will bind, then you will have less absorption. So here there is an inverse relationship between the absorption and the concentration of the whatever antigen you have in your sample. Okay, so that's why it's called competitive, because they compete. So there are two types of assays. You either bind the antibody, and then you have your three antigens from your sample, and you have the hapten that is bound to the enzyme. So these are competing with each other, the free and the labeled, linked or labeled. So again, when you have less antigen in the sample, more of the label will bind. This is the antibody bound. You can have a hapten bound the surface. So you have your hapten, protein binds to the surface, and then you have a labeled antibody, so the opposite. Okay, you have labeled antibody that will either bind to the free antigen in your sample and get washed off, or bind to the antigen on your bound hapten. Again, inverse relationship. If you have a lot of free antigens in your sample, they're going to bind to the labeled antibody and they're going to be washed off. So the enzyme will be washed off, and then you will have less absorption. One thing you want to know is the hapten, the protein you use here is different than the protein you use to inject the hapten, because you don't want the antibody to bind to that protein here. You only want it to bind to the one epitope that you have for the molecule. So this red is very important to remember, that the hapten that you use in the assay is different than the hapten you use to generate the antibodies. This is an example of using aflatoxin detection. The more aflatoxin I have in the sample, the less I get of the linked or the linked enzyme or the marked epitope, then I have less absorbance. When I don't have any aflatoxin, I get maximum absorption, because all of the possible sites will be bound to the labeled hapten. Okay, very quickly, it's the chromatography. We covered chromatography. So when I am after purifying a particular antigen, sometimes I'm purifying antibodies as well. Then I will have the stationary phase with the functional group being an antibody. Your whole sample is going to go through. You have very specific binding to the antigen of interest. Everything else will be on top. Then if you want to collect, then you change your mobile phase. Remember, you enhance the strength of the mobile phase. either you change the pH or ionic strength, because then you disrupt the nonpolar interaction, or you can add free antibodies to the mobile phase, and then your antigen will come out. This is the lateral flow. So what you have here is basically you have a labeled antibody. So here you have a labeled antibody with a color. And then if you have an antigen, if you're looking at an allergen, if you're looking at specific protein, if you're looking for COVID antibodies. So what happens is that they will bind to your labeled antibody and they're going to move up. And they will be sandwiched with another antibody that is immobilized at the test line. If they are sandwiched here, then you see a color. Now, this is just an illustration, but this is the real test. So what you have here is your color-bound antibody. Then you move up to the test line. If you have an antigen, it will get sandwiched here, and you will see a positive one. But there is a control line to test that the strip is not blocked. You want to make sure that there's always this control line. Here you have antibody to the colored antibody. So basically, you have an immobilized antibody to the antibody. Then you always see that control line. You always should see a color. If you don't see a color, there's faulty strip. So you would always have that. And then if you have the antigen, you will see a color at the test. Okay, so it's very common test for field testing of GMO, for example, in food analysis, because we care about food in this case. It's a quick test. You just do it on the field, and it is an example of sandwich ELISA. The last one here that I have for you is Western blot. So what happens in Western blot, and why I have this here with me, is first you run a regular gel, and sometimes it's a 2D gel, you get spots of proteins on your gel. The gel is very fragile, so you cannot, store the gel in different solutions to get interactions happening. It breaks very easily, it's not made for multiple analysis steps. So you have to transfer the spots, the protein spots, onto a PVDF membrane. So it is a specific membrane that has hydrophobic surface, a very sturdy membrane that the protein will transfer onto that membrane. And how do we do that? We have a special unit to do it. So basically, you put on one side your gel, on the other side, you put the PVDF membrane. You close that somehow, and then you would just put it in here in the compartment, in a specific compartment. You can run 2 at a time. And we run a current, a voltage. Then the protein will only move to one side. Okay, from when you run the current, electrons are going to move negative to positive, so they're going to move only in one direction, and you want to make sure that you put it in the right direction. Anyway, you don't have to worry about that now, but this is what we do when we transfer. We call this Western blot. So you transfer, and you take that membrane, you put it in a container, you add a solution that has antibodies specific for one of your antigens that you're looking for. It will bind. You wash, you block, and then you put another antibody that is labeled. This antibody is specific to the antibody that held your antigen. Then you also wash, and then you add your substrate, and then you allow it to react. And then if you have that antigen, you will see a color at that particular spot. So it's very nice technique. Qualitative, you can do the screening before you do quantitative analysis. This is an example. In this case, it's a 1D. You don't have spots, you have one-dimensional separation. And here's an example looking for cork protein specifically. So on your blood, you only see one color band, specific in this case for cork. So that is just an example. And do you have any questions? I still have 2 minutes. I finished early. Should I start a new topic? No. Any questions? Okay, so on Wednesday, what I will do, because we have that get together and potluck event. So I have the review session on Wednesday morning in lab. I will start off by a lecture, and then we'll do the review. And then we come to class, I will probably need time to finish like 10, 15 minutes or 20 minutes to finish everything. And then we can have lunch.
Is this room open before, class on Wednesday? Like, is it possible we could like bring our stuff here? Or
I'm not sure, when you come in,
Yes.
there's a class, maybe you only have 15 minutes prior to the lecture. Yeah. Okay.
Will you be in lab?
Yeah, we're gonna be in lab, so please be there at 9:30. I'll start off with lecture and then I'll go to, here, I wanna show you something on Canvas. Don't forget to turn in any of your, oh, you have your quiz return. If you have any extra credit that you did, please turn it in by Wednesday. I want to show you something here. So if you go to week 15, all the way down to week 15, you'll find a document that has many questions. So try to look at it before Wednesday. It's really important if you're able to solve some of these questions so that we can have an interactive review session on Wednesday. And I feel like your quiz was brutal again. But it's okay, it's practice for the final. I don't worry about it too much, but here's your average. It was pretty low. 18.3. The median and the average were close, so that is good. We have the distribution. And we got the highest, 28 out of 30, which is good. So that's an A. And then the lowest was 9 over 30. It's okay, don't beat yourself up on that. You have lots of points to gain and do those extra credits. All right. Don't forget to do the, if you haven't done the student rating of teaching, SRTs, please do that. And that's it. You can, yeah, you have any?
Yeah, and we also have the student evaluations for the TAs. If you haven't gotten a chance to do that, we would really appreciate it. Thank you.
Yes, please. It's important for our TA to hear from you as well. All of our TAs and our wonderful lab coordinators.
What was the product in the super?